Hello, everyone. Thanks for coming this morning. We're looking forward to having you all today. And I want to introduce myself. I'm Karen Odkins with the California Department of Fish and Wildlife, and I'm one of your CCHAB co-chairs, along with Jamie. Go ahead.
Hi, I'm Jamie Smith. I'm a senior scientist at SWRCB, and I'm one of the co-chairs as well. I think Sarah isn't making it right at the beginning today. She's our third co-chair.
Yeah, awesome. Well, we will jump right in. We have an exciting agenda for you today. Our theme is bioaccumulation. Oh, Sarah just joined. Hi, Sarah. Do you want to introduce yourself? I don't know if she can hear me.
She might not have heard you because it was
Yeah.
marked as pending when you had said that.
Oh, okay. Hey, Sarah, do you want to introduce yourself quickly?
Oh, sorry. Yes, this is Sarah Ryan, Environmental Director for the Big Valley Band of Indians and also co-chair of CCHAB.
Awesome. Thanks. Okay, great. So today, as we often do, we will have our updates from the various regions in our FHAB program. I update first, then we'll have a break at 10, and then we have a bunch of really exciting presentations for you today with the theme of bioaccumulation. And so I look forward to introducing those folks one at a time later, but let's get started on first with our FHAB programmatic update for the statewide water boards folks. Marissa, I think you're doing that one today. Is that correct?
Yeah, I'll be helping out today.
All
Hey,
right.
folks. Yeah, so I'm Marissa Van Dyke, the State Water Board in the Freshwater and Estuarine Harmful Algal Bloom Program. We implement this statewide in close coordination with all the regional water boards across 9 different regions. Take the next slide. Yeah, here's the map for you all. So our program has 5 full-time staff highlighted here in green, covering the North Coast, the Central Valley, and the Lahontan region. And I'm fortunate to have some volunteer staff at all the other regional boards, and we'll be hearing from most of them today on their updates that's been requested. Next slide. All right, so our program, with their limited resources, we focus primarily on incident response. So that's that short-term and long-term response to bloom reports that are submitted by the public or many of the CCAB members here today. We have an online reporting form on the HABs portal. I'll link it to the HABs portal. It houses all of our centralized resources, also produced from the CCAB members. So it's a really great tool to have. And as we can, we prioritize some like targeted monitoring, particularly since 2018, we've been hosting the pre-holiday assessments. So these are assessments that occur shortly before the major summer holidays, starting with Memorial Day weekend, then for Independence Day, Labor Day weekend, and then depending on the need, and if there's still an advisory up, we host a late fall assessment also if recreation season is still open at some waterways around California. So if you're interested in participating, we really encourage you to message us at the Sign Hub reports at water boards. I'll have that email in a moment shared there. And no experience is necessary. We can provide training and sampling kits and walk through interested parties on how to sample and to participate in the pre-holiday assessments. And that QR code takes you to a web page that has all sorts of handy resources and even some training materials. So if you're interested, please reach out. Next slide. All right. And now with in some places, unfortunately, cyanobacteria have already started occurring in some of the lakes and reservoirs. We also want to keep an eye out on rivers and streams. And in particular, if you think you may be observing a cyanobacteria bloom that forms these toxic algal mats, please keep in mind that CCAB Network updated their guidance for addressing benthic or these toxic algal mat kind of blooms. And now this will be their second year of implementation of this revised guidance. And all of the tools here are easily accessible following this QR code. There's customized advisory signs, a flow chart on how to start tackling the monitoring in the field, and also pre-recorded videos on some of the key pieces of the procedures in the field. So if you're curious about implementing this, or if you want a refresher, please reach out to us. We can assist you. Next slide. All right. And with that, as I mentioned, lakes and reservoirs are starting to have some blooms. So if in your jurisdiction or your area of interest, you have some larger lakes or reservoirs or really wide channels, perhaps they can be seen by satellite imagery. So just wanted to list out some of the useful satellite imagery tools that we have. This QR code takes you to the lead one that we've had up and running for a number of years. The FHAB satellite imagery tool that provides real-time cyanobacteria detection using that CI detection, not just chlorophyll, that could be also green algae. So that presents about 300 waterways across the state. And keep in mind that it does not see shorelines. That's, can cause issues there. So you do start seeing some cyanobacteria detections in these large lakes and reservoirs. That means that there's likely something else occurring on the shorelines. It's great to go out to those recreational areas and do that field verification and verify with your eyes if there is a bloom and to start conducting monitoring. It's just another tool in our toolbox to learn if there's a sign of bacteria detection besides going out there in the field. But we really encourage going out in the field. And so please take a look at that. If you're curious on how a waterway has been doing, this doesn't seem to have a bloom right now, but in past years, there's also status and trends app listed here second. It has 5 years of data that's summarized in some statistics that just tell you, some key points about if there's any cyanobacteria blooms in the past, and some of it's relevant, magnitudes, duration, that kind of thing. And you can look at that by county level and individual waterway too. And of note, USCP's HAB forecasts are up. They started that just, I think this is the second week that they have their forecasts back up. It's a website that also is based off of satellite imagery. Again, for larger waterways across our country, you can zoom into California and take a look. They provide weekly forecasts there. And next slide. All right. And so before I hand it off to regional boards to update, and also all of our great partners in CCHAB for their updates, wanted to inform you all on some of the monitoring projects that we'll be implementing this year as part of the FHAB program across the state. The first two in particular, these will be statewide implementation focused on benthic cyanobacteria, again, those toxic algal mats. We're going to be launching our year one of a pilot to start developing an ambient monitoring program to monitor for these benthics. So which site selection has already started. So if you are interested, we have capacity to bring on another partner, so please reach out. We'll also be comparing the benthic cyanobacteria sampling methods that are available from several entities across our country, and learning a little bit about their strengths and weaknesses, and learning if perhaps CCHAB procedures could be updated in the future. And we'll be, of course, pursuing incident response and supporting everyone that reaches out, and also hosting that pre-holiday assessment. Next slide. All right. Yeah, so please reach out if you have any questions. We encourage you to contact us if you'd like to partner with us. Thank you, Karen.
All right. Thanks so much, Marissa. A lot of exciting things coming up on the horizon there. And now we'll jump into our regional FHAB updates. As always, we put Region 1 on the hot seat first, North Coast. Kathy, Mike, I don't know which one of you is planning on speaking today.
Hey, good morning. I think I will be giving the update this time, but I do want Kathy, if she's available, to say a quick hello. So, yeah, Kathy was hired a couple of months ago to be the new regional FHAB and SWAMP program lead. Kathy comes to us with a lot of experience in water quality and lab work. She was previously doing water quality related stuff in Peru, and then got some ecotoxicology experience in Canada. So we're very happy to have her on board. But yeah, Kathy, if you want to say a quick hello.
Yeah, can you hear me well?
Yes, we can.
Okay. Hello, everyone. I'm Kathy Davila. I started working at the Water Board 2 months ago, and hopefully next time I will be presenting for you. Hi.
Awesome. We're excited to have
Thanks. Okay. All right. So next slide, please, Karen.
you.
Yeah, so routine monitoring we have planned in 2026. This is pretty similar to what we have done in years past or our partners have done. Klamath Basin, the tribal consortium up there is monitoring for microcystins. Big Lagoon also monitoring for microcystins. And then the Russian River, Sonoma Water will be doing their usual cyano IDs and percent cover estimates. Next slide, please. We are also doing a second year of partner monitoring in the Klamath Basin, specifically looking at benthic cyanobacteria. So we're going to be asking our partners to implement the tiered benthic monitoring approach, which is kind of illustrated there, basically using spats to determine when you conduct visual assessments. And then when you identify a bloom, collecting that benthic mat sample. So this will be occurring at 12 stations again this year, and more information is linked at those reports there. Next slide, please. We also have a lot of other partner monitoring going on in the region. So we will be supporting, hopefully, Blue Lake Rancheria to do tiered monitoring on the Mad River, the Baduat. We're hopefully helping the Potter Valley tribe as well as the Wiyot tribe to do tiered benthic monitoring on the rivers listed there. And then Sonoma County Environmental Health has dedicated funding to do tiered benthic monitoring on the Russian River. So we're very excited about that. And there are many other partners doing a lot of monitoring too, but we're kind of showcasing these since they're doing the full tiered approach. Next slide, please. Oops. Pre-holiday assessments, these are the water bodies listed here. So we're doing 7 again in the region, and obviously don't have any results yet, so stay tuned. No postings yet, but we did have one marine bloom or suspected bloom in Crescent City Harbor up in Del Norte County, but it seems like that has since dissipated. Next slide. No illness reports yet, so we're good there. Next slide. We will be doing our partner trainings again this year, and I will be sending out an email to our regional partners with these final dates and times. But we'll be doing that virtual component in early August at the time listed there. And this will be followed up by 3 options for field trainings in the Scott, South Fork, Eel, and East Fork Russian Rivers. So we hope to see everybody out there again this year. Next slide. Yeah, and always wanting to thank all our partners that we have been working with. I love how this slide gets more and more crowded and harder to add names in because we're just adding partners every year. But we have completed our annual check-ins with these partners as of this winter. And we have distributed all our emergency response or pre-holiday assessment or tiered benthic monitoring kits to them as well. So we're getting ready for hopefully not too intense of a season, but we'll see. Yeah, I think that's all we have.
All right, you guys are busy. Thanks for sharing.
Thank you.
All right, next we have Region 2. So I guess we'll start off. I'm going to be presenting for East Bay Parks. Today they had an all-staff meeting, so they were not able to make it, but they wanted to give an update on what water bodies that they manage are currently having advisories. So as you can see, Lake Chabot has a danger. Quarry Lakes also some dangers and cautions, and even a no advisory. Del Val has a caution. Shadow Cliffs has a caution. Temescal caution. Contra Loma caution. Lake Anza no advisory. Yay! And Big Break also excitingly no advisory. They are going to be doing some treatments this year. It looks like they're going to do some aluminum treatment and phoslock treatment at the end of April at Lake Temescal, and Lake Anza will also have treatment at the beginning of May. So that's their quick update. I'm sure they'll be here next time. They usually come and update us a little more on how some of those treatments are going. And Martha, did you want to do some other updates for this region? Marissa, sorry, you're on mute if you're trying to talk.
Again, sorry. Technical difficulty. Yeah, so Regional Board 2, this covers the San Francisco Bay region, just to familiarize everyone. And they are getting ready to coordinate with all the different entities that monitor and respond to with marine blooms and freshwater blooms in San Francisco Bay. There's a large regional monitoring program that they coordinate with and many different parties. So with that, they also are planning to do some sites with partners for the pre-holiday assessment. That'll be a little bit in the Napa River area in the northern part of the bay, and there's some parks with some stream access, and then support East Bay Regional Parks as they need. And East Bay Regional Parks has a really robust, comprehensive monitoring program where they're keeping an eye out on all their lakes with public access, especially with dog access. And we really appreciate their efforts in monitoring the East Bay. Okay, I'll hand it back to you, Karen.
Thanks. All right, on to Central Coast. Melissa is in the field today.
Right.
Yeah, did you want to go for her, or I can if you prefer?
Yeah, I can give an update. So yeah, Melissa is on the field, and she wanted to provide an update that they will be also monitoring for the pre-holiday assessments at routine AB monitoring sites for general monitoring for physical habitat, chemistry, that kind of thing. They'll be covering 6 lakes around the San Luis Obispo area, so it's north of those green dots. That's going to include lakes named Lopez, Laguna, Santa Margarita, a little bit more closer to San Antonio there. A little bit south of that is Oso Flaco, which has connection to the coast, a little bit marshland. They'll be monitoring there as well. And then in particular for Lake San Antonio, which is north of San Luis Obispo. It's a long, narrow lake. It's used for some small water systems for drinking water. I believe also irrigation. They've had some concerns over the past few years of some water quality issues and some detections of cyanobacteria blooms, wanting to understand a little bit more about those dynamics that are happening there, also causing some fish kills. So it is a site that we're going to be implementing a lake study this summer, targeting the warmest months, starting in May. So they'll be going out by boat using BB FICO Pro. Sorry about sharing my screen here, but the FICO Pro will be using its deployable tool, a measurement device that you can do depth profiles from a boat and understand a little bit more about the phytoplankton community that's occurring and do some other water quality measurements, also phycocyanin, pigments measurements, turbidity, water temperature, so forth. We'll be doing some other in situ measurements as well to characterize at multiple depths and all across the lake to understand a little bit more of those dynamics. So we'll look forward to learning a little bit more about the study. And that's it for the Central Coast Regional Board. Thanks, Karen.
Okay, Region 4, LA. Emily, I don't know if you're on here.
Yeah, so Emily is out on leave. We'll look forward to seeing her back in the fall. So yeah, Charlie and I will be supporting the LA region on any event response necessary there. We have been coordinating also with some partner entities in the Los Angeles area to do some pre-holiday assessments at some parks with lake access. So that'll get started up in May, and any outcomes of that will be published on that ad now. Thanks for having that little clip right there.
Oh, all right, Dana, you're up for the Central Valley region, that big one in the middle.
Good morning. I'm Dana Schultz with the Central Valley Water Board. We have a couple advisories currently in our region. So there's a caution at Contra Loma, which you saw from East Bay Regional Parks District routine monitoring update. There's a caution at Eastman Lake based on a bloom report. And we have an algal mat alert at the North Table Mountain Ecological Reserve based on CDFW routine monitoring. And then looking ahead to this coming summer, we're going to continue partner monitoring with Restore the Delta and Tulare County Environmental Health. We're going to participate in the FHAB program pre-holiday assessments, and we have some continued partnerships and some new partnerships, and also some monitoring groups in the region that sample within those windows and share their results. And then there's some other staff and other units in our office that have continued ongoing work in the Delta and Clear Lake that have HAB components.
Thanks. Sarah, did you want to give an update?
Yes, we've been continuing our winter monitoring of the Clear Lake, which is basically a once a month monitoring at 10 sites instead of the normal 20 plus sites. We've been doing mostly sending off for qPCR analysis to see if there are toxin producing genes for microcystin and potentially other toxins, depending on what we've been seeing in the lake via microscopy, and have actually seeing no detect of some of the toxin producing genes. This month for March, we haven't conducted yet for April, but for March we were out there, and it was the first time that we started to see a little bit of cyanobacteria. Mostly it's been green algae and diatoms. So we're seeing a little bit of Dolichospermum and Limnothrix in a few of the sites, so just kind of keeping an eye on that. We'll continue the monitoring and move up to twice a month monitoring, maybe by May or so, May or June. Also, there is a phosphorus, let's see, there is a treatment that's going to be happening in April. I believe it's let me find it. Through the Blue Ribbon. So there are a bunch of mitigation measure pilot projects for HABs in on Clear Lake that is being funded through the Blue Ribbon Committee and ultimately through the Natural Resources Agency. And one of those pilot projects is happening soon. There's a phosphorus mitigation demonstration that's starting April 13th. It's a project led by Utrafix and is a project that was approved by the Clear Lake Blue Ribbon Committee. And they're going to be applying Utrasorb in one location in the upper arm of Clear Lake in the off the shoreline out of Lucerne Harbor Park. These are areas where we do see in terms of toxin analysis of the middle of the arm and also on the shoreline, we have seen the caution, warning, and even danger, mostly in the caution the caution levels over the years. Anyway, they're going to be doing the 14,500 pounds of phosphorus is expected to be mitigated in that area. So again, a 400-acre treatment area located about 2 miles offshore. So that's going to be happening. There's also been an installation of to do hypolimitic oxygenation in the Oaks arm of Clear Lake, and that is a study that is being implemented by UC Davis and the Tahoe, the Turk, but they spent a lot of time on Clear Lake as well, and they are they're already in the midst of installing some components of it. There's a few other mitigation measures for HABs that have been approved for funding and will be happening. We're trying to figure out a way to do these pilot studies in areas that won't impact other studies so that you can get a real, understanding of is it working or not. So some of that detail is still being worked out. But over the next year and a half or so, I think we're going to have several of these things happening, and including nutrient removal and other types of studies that are going to happen. So other than that, we have been, there have been a couple of fish die-offs on the shoreline, which have been attributed to dissolved oxygen in the last couple of weeks that have been in terms of, it's mostly shad and the vertical profiling in those areas. It's been really isolated to do just 2 spots, so probably not HABs related because we're not really seeing any HABs in the lake at the moment. And I think that is it for Clear Lake. Thank you.
Okay, thank you for the update, Sarah. You guys are super busy as always. All right, Region 6, Lahontan, Sabrina.
Good morning. Can you hear me?
Yeah.
Hi, everyone. I'm Sabrina Rice. I work for the Lahontan Water Quality Control Board, Region 6. Yeah, next slide is fine. Thanks. So yeah, we're going to be participating in incident response and partner monitoring. So, we'll respond to incidents as they come in. And this year we've had kind of 1 report so far, but it was just some filamentous algae in a stormwater basin. And then partner monitoring, this is pretty new actually, but we just discussed with the League to Save Lake Tahoe a couple days ago that they are having some just citizen scientists or citizen volunteers going out in the fields, and their main goal is just going to be to informing the public about aquatic invasive species, but they are also going to try to get them to do some just general visual algal monitoring around the Lake Tahoe beaches that they're going to be at, and that yeah, it's kind of exciting. We'll have just some more general eyes on the lake there, looking at algae. Next slide. And then pre-holiday assessment monitoring, we'll be doing this again. We typically monitor around 30 water bodies per holiday assessment. For the most part, we do the Independence Day, Labor Day, and then the post-holiday assessment because we often see a later bloom season in our region. Next slide. And then we'll be participating in the statewide special studies. Marissa had mentioned these a little bit already, but there's the pilot benthic monitoring study and the benthic monitoring comparison study. The details of this aren't fully fleshed out yet, but it looks like we'll at least have 1 to 2 sites that we'll be helping with. Next slide. And as always, thank you so much to all our partners. We could not do all the monitoring that we do in our region without you since it's such a large region. And that's it.
Thanks, Sabrina. Oh. All right, Colorado River Basin. Jeff. Marissa, is Jeff on here?
Sure.
Marissa, would you cover? Thanks.
Yeah, now hearing Jeff, I'll provide a brief update. As part of the FHEB program, we also try to fund as much as we can some special studies. And we had a solicitation period late last year, and we're able to award some funds to this region, primarily for special studies focusing on the Salton Sea. So I wanted to note also that Jeff is partnering with the county, Imperial County, to monitor Weiss Lake around the holidays as well. It's a popular recreational park. Again, Imperial County, so like transition back to Salton Sea. Salton is a saline lake to orient folks. And the special study will be in partnership with Department of Fish and Wildlife, particularly with Barbara Berry, who will be hearing from a little later today on an associated project. So the monitoring will be focused in a habitat that's been constructed in the southern end of the Salton. It's referred to the Salton Seas Species Conservation Habitat. And it's a large-scale restoration project designed to create aquatic habitat and provide ecological benefits to the southern end of the Salton Sea. And there's been recently constructed aquatic habitat ponds and that have water in it that's been diverted from the New River that comes in to the Salton Sea. And upon some monitoring, late fall, winter, there was observations of some cyanobacteria blooms, primarily benthic. And the Salton Sea itself has just, widespread prevalence of HABs almost year-round, both planktonic and benthic, and also some marine blooms. It's a unique system being so saline. So it's great to keep an eye out on these constructed ponds as well. And so the monitoring will just start focusing on a few of the ponds to get some baseline conditions to understand a little bit more of what's happening in the habitat and inform the restoration activities. So looking forward to that project with Jeff and the Department of Fish and Wildlife. And that's the extent of the update for that region, unless there's any other partners that would like to speak.
All right, Santa Ana, Kirk.
Can you hear me?
Yes, I can. Thank you.
All right. Good morning. Kirk Larkin with the Santa Ana Regional Water Quality Control Board, the FHAB coordinator for the office. Next slide, please. So it's pretty quiet right now in our region. This is a snapshot of our HAB report map, sort of focusing on our area. The only data that you really see here is the gray pins for Lake Elsinore. Lake Elsinore provides us some monitoring, and we post those on our webpage. As you see that the most recent data goes back to January. So right now we don't have any new bloom reports or incidents that have triggered. So it's so far pretty quiet. Next slide. Taking a look at the HAB satellite analysis tool, I did focus on a couple locations. For example, Lake Elsinore in the southern portion of the region, Lake Hemet out towards the east, as well as Big Bear towards the north. If you notice the pixel coloration, we are starting to see indicators of potential cyanobacteria blooms that are popping up. In the next couple of slides, I'll focus in a little bit more details on those. Next slide. So here's a snapshot of Big Bear Lake. As you notice, looking at the lake on the left in the image, the pixels they're basically showing there's no presence of cyanobacteria at the moment, based on satellite imagery. I know the slide's a little bit busy. If you look at the top right corner, you see the trends for cyanobacteria blooms that have occurred over the last couple of years. Typically, Big Bear does get a cyanobacteria bloom, but it's later in the spring, summer, potentially continuing on into the fall. So far, so good. Hopefully the conditions last like this for a while. Next slide, please. Here's a snapshot of Lake Elsinore. Again, these satellite imagery data was taken just recently. So as you see, there's a trend in the last month or so. There has been an uptick in cyanobacteria that's been occurring. We haven't received any data from Lake Elsinore. We haven't been out conducting any monitoring yet, but it is a location that we will be conducting pre-holiday assessments. And if conditions start to get worse, we'll proceed with some visual and potential sample collection as well. Next slide. And then, as I mentioned earlier, here's Lake Hemet that's located in our eastern portion of the region. Smaller lake, but it is popular for recreational fishing. It's a campground that's near there that people come out along the shore. So an area that has high interest in public use. So it's a lake that we do look at as part of the pre-holiday assessments as well. Similar to Lake Elsinore, you see that there's a bloom that's starting to occur. Again, in the last month or so, you see there's an uptake, looking at the blue graph for the modified cyano index. And have not received any bloom notification for the lake yet, but again, it is on our radar to take a look at if conditions start to get worse. Next slide. And then just as a summary of our pre-holiday monitoring, we are looking at collecting samples at Big Bear Lake, Lake Elsinore, Lake Hemet, as well as monitoring two local creeks that have a lot of recreational use in summer, Mill Creek and Lytle Creek. Mill Creek and Lytle Creek will probably do visual assessments prior to the Memorial Day holiday, but it typically doesn't receive any type of benthic blooms or any other indicators till later. So more than likely those samples will be July, 4th of July, Labor Day, and post-holiday. And that's all I have for Region 8.
Great, thanks so much. Last but not least, we have Region 9. Carrie?
Yeah, so Carrie is out in the field today. We have a lot of projects up and running, also for their ambient monitoring program under SWAMP. Of note, just wanted to say her colleague of, oh gosh, 10, 15 years, Debbie Woodward, had retired and late last year, and a new team member has joined, and we'll hear from her. It's Kate Becker has joined the team down in San Diego. Just wanted to welcome her. Next slide. All right, so Carrie prepared some slides to give us all an update on what's going on. And we'll just go straight into the instant response and also their work to notify local water managers of satellite imagery alerts as well, because we've been implementing the World Health Organization's recreational alert threshold in their satellite notifications. The next slide. All right, so this is a summary of some notable incident responses they've had this calendar year. Cedar Creek Falls, it's a really popular waterfall at the end of the trail outside of San Diego. It's a really beautiful area. Though the most ponded area has had a bloom also in the winter months, particularly with microcystis and microcystin detections and the warning level. And then notably with the rain that occurred, they were able to depost the area in March. So that was great to hear, and it's still deposted from what I understand. So it's great to see that change. And then another notable bloom that's occurring is in Waffle Lake, and that is dissipated, and that's now deposted, and does have some amount of plankton still in there, but no toxins at the moment. Next slide. All right, and Carrie really wanted to highlight their plans for the holiday assessment starting in May. They have a whole host of sites, and in particular several new partners that have come on board, which she wants to highlight in a moment. But this is a list of their places that they will be partnering with to sample. So Cedar Creek is, again, what we just spoke to, and then they also focusing in on Wheelan Lake and Polis Lake, which also have had blooms in the past. Also looking into Mission Bay, this is some recent shellfish monitoring, finding microcystins in the tissue. They will also be focusing on some other historical sites, Eagle Scout Lake and Treat Lake. Again, historical sites with blooms and some nutrient impacts as well. And then down to the bottom, they'll be partnering with Department of Fish and Wildlife, in Laguna and Newell Lake, and then also moving into San Diego River and Mimosa Slough with partners as well to do additional monitoring for both planktonic and these benthic or toxic algal mappings as well. We're looking forward to this expanded network of partners. Next slide. All right, so just wanted to highlight the new partners and old. Really appreciate the San Diego River Park Foundation for being such a continued member for many years for the holiday assessments, and new members include Groundwork San Diego and multiple sites, and also the Friends of Mimosa Slough. So we're going to be getting some more coverage into the estuarine environment, which will be really great because there's also a bioaccumulation monitoring program along that area, and that data will likely be complementary and help inform some the monitoring questions helping there. Next slide. So of note, also San Diego region, so Carrie and Kate will be supporting the statewide special study looking at ambient benthic mat conditions. They will be selecting a site in one of the streams here. This map is just a snapshot of the tool that we're using, where we plotted where the USGS stream gauges are, but not just where they are, but also which ones are producing data in the last few years. If anyone's interested in this, maybe we can make this a little bit more broadly available. It's useful to know where these are occurring. And we also plotted our historical benthic advisory sites for the past 5 years on here to understand the potential areas to target, particularly within a good amount of driving distance around the regional port offices. So that's what's denoted here in the red. So they'll be keeping an eye out on some of these streams and determining where to go to do that ambient monitoring study. And this will start in June, run through the fall, going twice a month and implementing our pilot study, looking at benthics and also the environmental context that may be supporting this benthic growth as well. So looking forward to all these studies. Thank you, Karen.
All right. So I wanted to ask if anyone else who I did not call is on here and would like to give an update. Feel free to just unmute if you are in that position. And if not, does anyone have any questions for these folks? We're running a little ahead of time, which honestly never happens in this section. So it's a rare opportunity to be able to ask questions if you have any. Of course, you can always email people too, but.
Yeah, I have to second that. We normally run over time.
I know.
This one's unique. We actually elaborated about some of these studies. If anyone has any questions, please use your hand. Why don't you mute yourself?
Well, given if there's no questions, we can have a little bit of a longer break. I don't want to throw off the rest of the meeting with starting things super early. So let's take a break and we'll come back at 10:10 and I will share that so you guys can just see that. So you get a nice long 20-minute break. Enjoy, get some coffee, check some emails. We'll see you soon. Hey everyone, please gather back around. Sorry, I just got started like a minute before we said it. I don't know if people were, how long time people are.
People are pouring their third coffee before they run back to the
All right, sorry. Marissa, do you want to start over? She had a quick for us before we jump into the presentations.
Yeah, well, now that we're officially coming back from break, so to put in a quick plug for the tribal training series. I got some direct messages asking about this, so I thought I'd highlight this once more. I know we've been sending this out on the listserv many times to many different listservs, so sorry about that cross-posting there. Wanted to highlight that registration is open for the April and May events. So April 23rd and May 27th are the next two events focused on monitoring design and then sample collection. And if this sounds interesting to you, the past material is available on YouTube. There's recordings and the slide decks. They are made on purpose to have lots of material and resources like reference cards. So please check them out if you're interested. And just a little background, this training series has been a long time in development with many partners. So the Surface Water Ambient Monitoring Program partnered with the Big Valley Band of Homo Indians, the Yurok Tribe, San Luis Rey Indian Water Authority, formed a tribal advisory committee there to help inform us on how to develop this training series to make sure that the information is most useful to all parties. And this is open to tribes, so please feel free to share this information. I'm putting the flyer in the chat with a download PDF, not on Google Link, so hopefully that's most accessible to folks. And yeah, looking forward to any new attendees. Thank you all. I'll hand this back to Karen and Jamie.
All right.
Include that.
Thank you, Marissa. All right. Great. So our first speaker for today is Misty Peacock. She's coming from the Northwest Indian College, and she's going to give us a talk about freshwater toxins and marine harvests. We're so excited to have you here. Do you want to go ahead and share those slides? You're muted as well. But...
So it's not going to let me share, actually, unless I quit and reopen.
Oh,
So,
okay, I can share the slides for you.
Yeah, I'll just tell you when to switch, and I appreciate it.
All right, that's why we got the backup. Opening, give me 1 second here.
Hi, everyone. I'm Misty Peacock. I am from Northwest Indian College. I have a bit of a cold, so I apologize for that. I'm actually in California. It's our spring break, and I am in Arcata, California. So I am visiting my state of birth for a little bit. Thank you so much for inviting me to give a talk. I'm going to talk about the transport from rivers to shellfish, the freshwater toxins and marine harvests. And the work that I do is on the tribal lands of the Lactamish people, which is people of the sea. So we are associated with Lummi Nation, though we are a 4-year degree granting tribal college. Next slide, please. And so one of the things that we do is we have a T Center for Community Marine Research. And so the work that we do is really focused on identifying community research projects. We work with tribes up and down the west coast of the United States. We support tribal nations in food and data sovereignty. And we engage in community knowledge exchanges within those tribal markets. And so I'm pointing this out because I really want to talk about freshwater toxins and their impacts on the marine environment. Next slide, please. So our research lab, we have 8 full-time researchers, and we also have, during the academic year, usually about 5 student interns. In the summer, we run a really robust internship program, and we have up to 15 summer intern students. These are all undergraduate students. We also have graduate students working in our lab. And so all of the research that we do takes into account those Indigenous students and their impact with what they're going to learn is take it back to their tribes and work in natural resources departments or go on to graduate school with that degree. And so here's just some photos of people in the lab or students and doing some of the work that we do. We are sampling net tows in the marine water. It's kind of stormy in Washington a lot of time. That's at Gooseberry Point, which is on Lummi Nation. We are harvesting clams during low tide. And we also have a fully functioning LCMSMS, liquid chromatography mass spectrometry. And so that's how we run all of our biotoxins. And the projects we focus on are seafood safety and water quality monitoring. Next slide. And so I feel like I'm preaching to the choir, but cyanotoxins are found persistently at the land-sea interface. A lot of people think that they're just a freshwater problem, but we really try and work with our students and our community that everything that happens upstream eventually ends up downstream. And so this is a review paper by Ellen Preece from 2017, and this just shows all the places in the world where there's this persistent cyanotoxins at the land-sea interface. The harmful concentrations have been found in invertebrates, fish, mammals, and humans. Some of the seminal papers have come out of California where cyanotoxins were impacting shellfish, we're impacting birds, and we're impacting marine otters. And it can absolutely disproportionately impact subsistence harvesters. And so subsistence harvesters are people who are harvesting not just for recreational commercial purposes because it feeds their souls or it feeds their families. And so a lot of these people who are harvesting subsistently are actually ingesting a lot more shellfish than the average adult human would. Next slide, please. Oh, sorry, that's an arrow pointing where I am. I'm in that corner of Northwest Washington. All right, next slide. So how do they enter estuarine and marine environments? So that top right corner is a picture from Avery Tatters when he was down at in Southern California, and you can kind of see those filaments of cyanobacteria. The bottom picture, of course, is Florida's toxic algae crisis, and so this was in 2019 when Lake Okeechobee had to be released, and a large amount of microcystis was flushed from that lake into the seawater, and they had pictures of manatees and humans who were swimming in this water and covered in this green cyanobacteria. So they grow in freshwater, but they often can get flushed into estuarine and coastal environments. They actually last a lot longer than a lot of people thought. And so we anticipate that they would probably burst within about 24 hours. There are some studies that show that they can last upwards of 72 to 96 hours in a saline environment as an intact cell. And when we have high tides or increased storms or a lot more river water input, we're seeing these flush events get into the marine environment a lot more frequently than they did in past centuries. Next slide. So this is from my hometown. So Lake Patton is that picture on the left. This is a photo from 2007, and you can actually see that kind of blue-green algae color at the water. This is actually a microcystis event. The photo in the middle, the micrograph in the middle is taken from the Rafe Kudelis lab, and you can actually see that colonial microcystis. It's about the width of a human hair. They're very small, but when they are in very large quantities, you can actually visually see them on the surface of the water. And then that bottom picture is from Weiser Lake in Whatcom County, and this is where the bulk of our microcystis and microcystin toxins that get into the marine environment are coming from. And the reason that we know this is that we actually have a study where we're measuring that kind of outflow from Weiser Lake into Weiser Lake Creek, into a larger river called the Nooksack River, and then into that coastal environment. And so we routinely see these freshwater cyanotoxic events. I'm talking mostly about microcystis and microcystin, though we often see other cyanotoxins like anatoxin and cylindrospermopsin. Next slide. And so this is a poorly done drawing in PowerPoint showing that kind of upstream to downstream impact. And so we in Whatcom County, where I live in this Northwest Washington, we have a lot of farm and agricultural runoff. We're actually produce about 95% of the United States raspberries in Whatcom County. And so there's a lot of nutrient runoff that happens. We also have a lot of lake input of that cyanotoxin. And then there's urban runoff. We're in a city of about 100,000 people. And so you can actually see that kind of upstream to downstream impact. And if you hit next, Karen. You can see that these can really impact decisions, especially if you're working with natural resource managers. Fishing and shellfish harvest access. A lot of shell fisheries are right at riverine or estuarine outputs, and the reason for that is because of the nutrients that are available. So we have a lot of shellfish growth. This can really impact shellfish openings and closures if you're testing for marine toxins, but you're not testing for cyanotoxins that may be in shellfish. Trust in the monitoring programs. The state of Washington and our tribal managers have joint control over shellfish openings in tribal tidelands. And if the tribes are testing for marine toxins but not for freshwater toxins, then there's a lack of trust that can happen with whether it is safe to eat these shellfish. And then future investments in monitoring when you're deciding where to put money as a state regulatory agency or as a tribal management group. Next slide. So, people in this group may be familiar with SPAT or solid phase absorption toxin tracking samplers. These are small bags of resins that are left in the water to monitor for biotoxins, and they can provide continuous monitoring for both freshwater and marine toxins. We put ours in embroidery hoops, and so you see in that top picture, that's kind of a close-up position of that pink embroidery hoop. There's a little bit of netting that's there where we have the spat resin beads. They're really similar to those St. Ives face washes where you used to scrub your face with the scrubby beads. They're about that size. And there's a small measured amount of this spat resin that is in these samplers. They measure dissolved toxins, not total toxins. So it's a semi-quantitative measurement, but it is cheap and easy to use. It's widely monitoring for both freshwater and marine toxins in the United States, also in Europe and Asia as well. So it's something that is used within the community to measure for toxins. We also place some on the buoy so that we can understand what's happening in the marine environment. That's about 2 nautical miles offshore. Next slide. And so just to place you where this research is being done and where we're sampling, the red outline on this map. So if you think about Washington State and you have kind of that bite out of the corner, the northwest corner of Washington State, that is the Sailor Sea. It is an inland sea. The Pacific Ocean, the Eastern Pacific is out there. Vancouver Island, which is part of British Columbia, kind of comes down. It's actually south of where we are in Bellingham. And you can see that faint black line that separates the British Columbia and Washington State. And then that top star is Bellingham. If you look down at that bottom star, that's the city of Seattle. It's about 90 minutes south of us. And about 90 minutes north is the city of Vancouver. So we're kind of right in the middle. And so I'm going to be talking about that bay, Bellingham Bay, which is right again at that little star that says Bellingham. And then the type of toxin monitoring that we do, we actually do all of our analysis on a liquid chromatography mass spectrometer, which we have at the lab. And that's my lab technician, Megan, as she's running samples for Microsystem. Next slide. So I'm going to show you a series of graphs, and I just want you, this is a monitoring over time, and so what I want you to take away from this is kind of the change over time as we're looking at these microsystems in lakes and in marine environments too. If there's an open circle, it means that we sampled, but we found 0 toxin, and these are on these spat samplers. If it's a big circle, that means that there's more toxin. So the larger the circle is, the more toxic there is. We're going to look at 2 lakes. One of these lakes is Lake Patton, and that was the one that had the bloom from 2007 of microcystis that we saw in the water. And the other one is Lake Whatcom, and that is the drinking water reservoir for the Whatcom County. Next slide. And so if you look at this graph across the bottom, you have time. So this is from 2020 through mid-2024. And then again, the size of that circle tells you how much microcystin we measured in the water. So the larger the circle, the more microcystin there is. And you can see that for Lake Whatcom and Lake Patton, what you do see is that there's kind of this persistent signal of microcystin in the water. So we routinely see this cyanotoxin in these freshwater bodies. Again, on Lake Patton, you can see the open circle where we tested, but there was no toxin. So there are some zeros. Next slide. Now we want to look actually at the output. So we know that there's toxin in the lakes, but we want to see, is that toxin actually getting into the marine environment? So we have two stations that we're looking at. One is Fairhaven Docks, and those red arrows actually show you the outflow of where those lakes come from. It's a very short distance. The longest one is slightly less than 2 miles. The one from Lake Patton is only about it's only about 3/4 of a mile long, so it's traveling from Lake Patton almost directly into the marine environment. So we have Fairhaven Docks, which is the Lake Patton outflow, and then we have the Schlein Buoy, which is about 2 nautical miles offshore. Go ahead, hit next. And so again, the larger the size of that bubble, the more toxin is present. And if it's an open circle, that means we tested it, but there's no toxin present. If it's a blank space, we weren't able to test that day. And so what we actually see, if you look at that top graph, the bottom graph is exactly the same from the last slide where you have those two lakes, but the top graph is the marine environment. And so we are seeing the transfer of this freshwater toxin getting into the marine environment. You see it at Fairhaven Docks, and you also see it 2 nautical miles offshore at the Schlein Buoy. And so you can actually see there's kind of these punctuated events where we have a large amount of toxin, except in 2023, where we had a whole bunch of negative zeros. Even though we saw it in the lake, we didn't see it in the marine environment. Next slide. We are also looking at toxins in the freshwater food web. I want to point out that we as humans know when we shouldn't ingest toxin and when we shouldn't be in a lake or something like that. But this picture is from Dr. Anthony Lapsansky, a researcher in our lab, and you can clearly see that it has a toxin closure sign right there, but that heron is not paying attention to it at all. And so some of the other things that we have tested for are the Nooksack River sediment. So we've tested for microcystin in clay. We've tested for it in frogs, which are tadpoles, or some bullfrogs or snails, and some of the freshwater species of insects. If you hit next. We can routinely see it in a lot of these organisms. And Dr. Lamsansky is actually working on toxin transfer into insects in the freshwater environment. And so that's his project, and maybe he'll be on at another date to talk about that. Next slide, please. But one thing that we do want to say is, one thing that we do want to focus on is not just the transfer of that toxin from environmental samples to environmental samples, from freshwater to marine water, but does that get into the marine animals? So we always want to protect human health. And so we're looking at if it is becoming an issue for people who are ingesting this seafood. And so this is the list of animals that we have tested. There's not enough time to go over everything, but you can kind of see they're broken up into groups. Again, we're focusing on subsistence and recreational harvesters, people who are not doing this necessarily for commercial harvesting. There's a really robust program in Washington State for commercial shellfish, but people are eating a lot of things more than just buying it over the counter. Next slide. And so the top one are the marine animals. The bottom one is those environmental and food web monitoring that we're testing for. And you can see that it actually expands. We're actually doing dragonflies and damselflies, as well as tadpoles and bullfrogs. And we actually had a lancet fish, which is a deep water fish that kind of washed up on shore. And we also tested for that. Next slide. And then some of those freshwater animals. And these, again, are just kind of what they happen to be dead on the shore. A lot of times is how we end up testing them. Somebody will come home back to the lab from sampling and say, I found a dead fish. So this is how we've got most of these samples. Next slide. So this is a complicated graph, and let me just situate yourself. So on our y-axis, we actually have the total amount of microcystin toxins. This is in nanograms per gram of tissue. I also want to point out that there is a break in that graph. So at 200 nanograms per gram, it goes from 200 up to 600 nanograms per gram. Microcystin is a toxin that can accumulate in the liver of animals. Most of these animals were tested as a whole animal, so it's one clam, it's one mussel, it's one fish. The only thing on that graph, you see kind of that high concentration in the duck liver. So these are all of the marine species, the marine animals that we have tested. So these are freshwater toxins now in seafood. So it's not just in the marine environment, it's actually in the seafood. So along the bottom, all of those different colors are different marine species that we have tested, and I've kind of put them in big groups. And so kind of the blues right there, the clams, some of the pinks are the fish, and then that duck liver is that really high sample up there in that brown color. I also want to point out that that red dotted line, there is no tolerable daily limit for ingestion of cyanotoxins, so freshwater toxins in marine seafood. And so what this is the World Health Organization's limit of 10 nanograms per gram per week. So this is the tolerated daily intake per week that you should have. And you can see that quite a few of these samples, and these are single samples, right? So this is not 100 grams of mussel. This is one singular mussel, exceed that tolerable daily weekly input. And again, just to point out that duck liver, this is a toxin that actually accumulates in liver, so we would expect it to be higher in something that has a liver because it's a hepatotoxin. But you can see that duck liver is up around, I think it's 646 nanograms per gram of microcystin. Next slide. And so one thing I do want to point out, and this is actually the number of animals that we tested. And so on the left of this table, you have the count. So we tested, for example, 39 butter clams, but we only tested 2 gooey duck. We only had 1 lancet fish, which happened to wash up on shore. And so what I wasn't showing you on that graph, and Karen, if you just hit next again, it'll pop up, is the percentage that are zeros. And just for clarity, I had taken them off. So you can actually see the ones that are in bold are actually samples that all had 100% zero. So they did not have microcystin toxin. And so this graph is a little bit skewed to show the animals that did have that toxin, but it doesn't really show the number of them that didn't have that toxin. The ones that are in blue are naturalized species. The one in red, the European green crab, is actually an invasive species. And so you can see that those species also sometimes had that microcystin in it. Next slide. So the consequences for human ingestion is unknown through seafood. As I said, that it's not there isn't really a tolerable daily intake limit for seafood. So this doesn't mean that seafood is unsafe, and it doesn't mean that closures are needed today. So we are not a regulatory body. That is not what we're saying. What we're saying is that there are monitoring gaps that exist, that freshwater decisions affect marine food, and that tribes should be invited to the table early and not last. So that we can make sure that everybody is able to understand this impact that is happening to seafood. Next slide. And I just have one more slide. And so we are a marine seafood safety lab. We focus on marine environment and seafood safety. And I just want to point out that we test for a lot of different toxins. And Karen, if you go ahead and hit next, it'll pop up. And so what we have here is the list of all of the toxins that we test for. On the left, yestotoxin, paralytic shellfish toxins, diuretic shellfish toxins, and domoic acid are marine shellfish toxins. And on the right are cyanotoxins or freshwater toxins. And you can see that some of these groups of food that we have tested for actually have kind of overlap in what is in both of those animals. So whether it is marine toxins or freshwater toxins, you can actually end up sometimes with the same seafood that has both toxins in them. Next slide. And so I just wanted to say Haishka, which means thank you, especially to our interns and our researchers who collected this work and the funding that we have, and just leave you with a few key takeaways that freshwater toxins are showing up in marine foods. It's across multiple species and multiple years. And the current monitoring programs really weren't designed for this. They weren't designed to test for freshwater toxins in marine seafood. And so communities have a lot to gain from early proactive monitoring, which is what we're trying to do with our tribal communities. Thank you. And I'm happy to take any questions if there's time.
Yeah, thank you so much. There is some time. Super interesting presentation. Does anyone have any questions? Probably take a couple. Happy to start us off. Oh, Sarah, go ahead.
Hey, Misty, that was an amazing presentation, and I'm just so thrilled to see the depth and breadth of the program there in order to protect human health and tribal health in particular. I work for a tribe, and we've done some cyanotoxin monitoring in fish and waterfowl tissues as well on Clear Lake, and I'm just really excited to see what you've done, and I'm going to contact you offline or online to just talk about it further. And the one thing I was curious about is that suggested limit of human ingestion. There is a suggested action level of 10 nanograms per gram in California as well. And I'm wondering what work that is happening in Washington or within the group of people you work with regarding subsistence consumption and an ingestion level for that.
Yeah, to my knowledge, there hasn't been any like dedicated work. There hasn't been any kind of regulatory studies specifically on that. We are working with the Washington Department of Health to look at how these cyanotoxins may be in seafood. So it could be moving that way. We do have Washington State does have environmental and drinking water levels for cyanotoxins, but to my knowledge, except for the recent FDA paper that came out from Jonathan Deeds with microcystins in mussels, there hasn't been a whole lot of work on an ingestion, a tolerable ingestion limit.
Thank you.
Great. James? You're
James?
muted, James.
Yeah.
James, you're still muted.
I'm
Now I
not
can
sure
hear
if
you.
I heard my name called or not, but I'll go ahead.
Yes, it's your turn. Yeah.
Misty, thanks. Great presentation. Yeah, I really like all the lab work you're doing and have lots of questions about it. But my one question is the very last graphic. You show anatoxin in the key, but I don't see it listed in being detected in any of those tissues. Did you not detect any anatoxins, or did you just not test for it?
We haven't. We haven't really seen, we see anatoxin in the environmental samples, but we aren't really seeing it in the shellfish. So we are testing for it, and we have not really seen it we haven't seen it in the shellfish. I'm trying to think if we've ever seen it in the shellfish. We might have seen one or two um, times, but we see it routinely in environmental samples.
Thank you.
And sorry, I do want to say that we have seen it in the marine environmental spat samples as well. So we not haven't necessarily seen it in the seafood, but we have seen it in the marine environment.
Okay. Thank you so much for presenting. That was super interesting. I have a question for you too, but we are at time, so I will contact you offline.
Yeah, and I'll put my email in the chat. So if people have
Perfect.
questions, I'm happy to answer them.
Amazing. Thank you so much.
Thanks,
Okay, up next we have Barbara Berry from my department, California Department of Fish and Wildlife, who's going to tell us about her Salton Sea study. Barbara, are you good sharing, or would you like me to share for you?
Yeah, you know what? Why don't you share for me?
Okay.
Great, thanks. Yeah, I'm Barbara Berry with the California Department of Fish and Wildlife with the Salton Sea program. Been with the department for a few years. Our focus is on species in the Salton Sea watershed, and we're involved with the state Salton Sea program, which I'll get into in a minute now that the slides are up. Next slide, please. So I'll be talking about our environmental DNA study that we did in the Salton Sea watershed. Just to give a summary, I'll talk about who we are at the Salton Sea program, the purpose of the study, Salton Sea background information, methods we used, very, very brief preliminary results, our next steps, and some acknowledgements. Next slide, please. So CDFW Salton Sea, our mission is to conserve, restore, and create habitats in the Salton Sea ecosystem in a manner that ensures the harmonic coexistence of species and human communities. Sorry, I had to read that off the page. That's our mission statement. We do this through environmental permitting, species monitoring and surveys, and we're involved with the California Salton Sea program, formerly Salton Sea Management Program. It was just rebranded. And the mission of that, I'll also read it off the page. The California Salton Sea program is made up of 3 state agencies, CNRA, CDFW, and DWR. These 3 agencies are advancing projects at the Salton Sea to improve air quality and provide habitat for birds along the Pacific Flyway. And I'll plug our 2026 annual report, which is now available on the website as of last week. And a lot of our work that we've done at CDFW is featured in the report. Next slide, please. So we did this study, started in mid-2025, went to January of 2026. It was collecting environmental DNA around the watershed to better understand our aquatic species composition in the area. This is due to a few factors that we found this study to be potentially very useful for us. The current conditions of the Salton Sea are changing very quickly, and I'll get into very briefly why that is later. We also wanted to get a better understanding of how our aquatic species composition is changing at the Salton Sea, phytoplankton, zooplankton, invertebrates, fish, etc. And we wanted to know what the harmful algal bloom community looked like in surface waters and potential correlations with wildlife health, and to kind of piggyback on the work that Jeff Garassi has done at the Salton Sea with algal mats and shoreline sampling. And very excited about his new study. We wanted to get baseline information on our constructed saline ponds that Marissa mentioned earlier. This will hopefully improve our ability to inform the construction and management of this project and other projects around the Salton Sea. There's also a lot of unknowns with our agricultural drains that flow into the Salton Sea that support endangered desert pupfish, and better understanding the aquatic community can also help us understand the pupfish. Next slide, please. So the Salton Sea, it's highly saline. It's a terminal lake, man-made, eutrophic, and it's in a very arid environment in the Colorado Desert in southeastern California. It was historically a very productive fishery and supported large numbers of fish-eating birds traveling along the Pacific Flyway. It's located in Riverside County on the north and Imperial County on the south. This picture here is from the satellite image, is from 2002. At that time, it was 35 miles long, 15 miles wide. And go to the next slide, please. So this is kind of giving you an idea of the watershed. It's Colorado rivers flowing through Arizona, going through Baja, and then north along the Alamo New Rivers up to the south side of the Salton Sea. It goes through a lot of urban and agricultural areas. For many years, the lake level was relatively stable because of these inflows, even though it's a very arid region, a lot of evaporation. And precipitation, again, is very low in the area, like 3 inches a year or something like that. And it doesn't really contribute too much to the lake level. Next slide, please. Okay, so this is supposed to give you an idea of how the lake shoreline is receding. Karen, I don't know if you could go to click on the bottom of the slide. It's a video. There you go. You see on the left side there. Yeah. So the red line is a mile marker. This goes between 2014 and 2026. The shoreline is reducing pretty quickly, and that's due to demands on the Colorado River, drought in the watershed, leading to water transfer agreements between agricultural to urban uses. The lake level has been declining for many years. This has led to the shoreline receding, and this has led to the salinity also increasing pretty drastically. It's been a salty lake for a while, but right now, based on Regional Water Board total dissolved solids concentrations they took recently, it's around 2.5 times as salty as the ocean. And salinity has doubled over the past 20 years. So salinity has definitely increased very quickly. And you can see in this video another major change that has happened to the area. And this, well, no, just leave it on this one. Sorry. The final, yeah. There's been a lot of construction in the area, and that's the species conservation habitat, which is constructing a series of saline ponds over the exposed playa in the Salton Sea. Next slide, please. It's just going through the video. There we go. So like I mentioned, this is converting Lake Playa to saline ponds. The ponds were designed to provide habitat for fishing birds and suppress dust from exposed playa. They started to get filled last April. We're very excited about that. And we wanted to use this environmental DNA study to get baseline information from the aquatic community to inform our management for species in the ponds. And now I'll get into the methods of our study. Next slide, please. So we collected surface water samples at several locations around the watershed, collected them in 1 liter amber bottles. Sampled bottles were placed in cooler after collection. We also collected water quality information from a YSI Pro DSS for dissolved oxygen, specific conductivity, water temperature, pH, total PE for chlorophyll A and phycoerythrin, collected Secchi depth, and additional field data was recorded with ArcGIS field maps. Next slide, please. After that, we filtered the samples. The most important materials we used in our sampling effort was the vacuum hand pump and the Wattman filters. Thank you very much to the State Water Board for supplying us with that. We used various bleach solutions to minimize cross-contamination, deionized water for rinsing and cleanup. After filtering, the filters were stored in centrifuge tubes on ice. The samples were filtered as soon as possible, between 1 and 4 hours. And then at the end of the day, were stored in a freezer at -10 degrees Fahrenheit until we were ready to ship them. And some of the sampling locations were pretty far away from our office, so we had to filter in the field as best we could. That's what's featured here. And now for the location sampled. So we collected samples across the watershed. All of the purple dots are samples locations sampled. In the Salton Sea, we collected samples in the mid-lake and shoreline areas. These locations were chosen based on minimizing freshwater inputs. the influence of freshwater inputs. And we had a lot of logistical challenges with boats, weather, and holding times. We sampled at the mouth of the New and the Alamo rivers in the south, and several drains and tributaries along the north side and southeast side. And we had several sites at the species conservation habitat ponds that's blown up in the corner there. And it's not featured in the satellite image, but the blue lines show where the ponds are. That's about 2,000 acres. And those are all wetted now. And I'll go into some of our logistical challenges with the study. Next slide. So much of our sampling was delayed by several months, unfortunately. And by the time we got out of there, it was a bit crazy with the weather. We had high heat conditions, a lot of rain, flooding, lightning. Our boat is a metal airboat, so we didn't want to be on the lake when there was lightning forecasted. High winds. It gets pretty dicey out there in the Salton Sea when there's high winds. We also had some issues with accessibility because the Salton Sea shoreline is decreasing so rapidly. Most of the boat ramps are they don't access the Salton Sea anymore, so there was limited access to the lake. The conditions around the Salton Sea can almost be like quicksand. It's you can sink pretty easily. There's also active construction at the species conservation habitat, so we had to work around the construction schedule. And our filtration equipment, we did have a little bit of a difficulty filtering in the field. And of course, we had issues with our boats. So next, I'll briefly go into our initial results. If you go the next slide. So we got our results as of last Thursday, so we haven't really had time to go through them, but I just wanted to give you an idea of what the results look like. We have thousands of data points to go through, but we're excited to see that I think one of the things that we were concerned about with this study, because the volume we were utilizing was not a huge volume, we were a little concerned that we wouldn't have a diversity of species, but we're definitely seeing that. We've seen fish and zooplankton, cyanobacteria, different types of phytoplankton. So we're seeing a really great distribution so far, and we're going to go QA and QC our results compared to blanks, and we have a lot of analysis ahead of us. Next slide, please. And this is just to give you an idea of the prokaryotic information that we're getting. This is just a small snapshot of the species that we're looking at. We're really excited to go through it. Next slide, please. So next step, like I mentioned, we're going to process the data. Our plan is to QA, QC the data and to summarize it and make it public, likely on our Salton Sea Hub site page that recently became public. We're going to finish our QAPP to update it with changes that we had to make in the field due to logistical challenges. We're also, fingers crossed, going to have some testing for chlorophyll A in the lab that we can compare with the results that we're seeing from our YSI total PE probe. And this will hopefully give us some insight into our pigment study that we've had to put on hold for a while at the Salton Sea, and to also compare to our results that we saw with the eDNA. Next slide, please. Lastly, just briefly go into acknowledgements. Thank you, Karen Odkins, our CDFW HABs coordinator, for making this possible. Dr. Susie Thoreau at SWRCB for giving me some initial pointers on how to do this. Tim Otten on Ben Genetics, thank you for making this study possible and processing all the samples and getting it to to the lab, Lindsay Metz at the State Water Board Swamp Program for also giving me some pointers about eDNA, and Carly Nielsen and Marissa Van Dyke for getting us our initial equipment to make this happen. Thank you.
Thank you, Barbara. Very interesting. I've never been to a lake with quicksand, so that's new. Oh my gosh, lots of challenges. Yeah. Well, we look forward to having you back when you have some more results to share and have thought through those. Unfortunately, we don't have a lot of time for questions here, so I did see Marissa pop something in the chat, and feel free if you guys have questions, either email Barbara or put something in the chat, and maybe she can answer it there. Thank you so much. And now we will be moving on to our next speaker. We have Corey Gibble and Rohini Joshi. I hope I said that last name correctly. Please feel free to correct me. From my agency again, CDFW, California Department of Fish and Wildlife. So feel free. Do you guys want me to share, or how do you want to do it, Corey?
Yeah, I'll try. Let's see.
Okay.
Let's see if I can do this. Okay.
I am seeing your screen, but not yet shared.
Okay. Is that?
Or sorry, not yet presentation view.
Okay. Can you see it?
There we go. That looks great.
Okay. I was like, it looks right for me. But I'm gonna just turn off my video for my talk because sometimes I have internet issues at my house. But then I'll come back on when we're done. Okay. So thanks everyone for inviting us here today. I'm going to talk about microcystin and nearshore feeding seabirds. So this is a little fun little project we've been working on. It's a retrospective analysis, and just keep in mind, these are just preliminary results. So we should have more later, but these are just sort of our thoughts right now. And it's of course me and Rohini, who you'll hear from in just a little bit, as well as Misty Peacock, who you heard from earlier, and Rafe Kudella from UCSC. So it's a collaboration between Cal Fish and Wildlife, UCSC, and Northwest Indian College. Okay, so our goals. So the goals for this talk really is to sort of set you up to understand where we're at now. So our new work aims to elaborate upon a previous paper from 2017, so a little bit of an older paper, but it's not a heavily read paper. It's sort of this, it's very niche. So I wanted to present a little bit of an overview of that paper and then discuss preliminary results for a future paper that's now in progress. And sorry, I'm struggling with seeing my own screen. Of course, I don't have to explain this to this group, but we're looking at Microcystis ruginosa, obviously a cyanobacteria with worldwide distribution, has major fall blooms, and it produces the toxin microcystin, which has many toxic variants, as you may know, and Rohini will go into a bunch of the variants that we looked at. It's a strong hepatotoxin, so that means it affects the liver, especially important when we're talking about birds. It's a regular occurrence in California freshwater systems, but as Misty sort of hinted to earlier, it's detected in the nearshore marine environments as well, and especially in Monterey Bay, and that's where our samples came from, so that's important here. So here's our paper from 2017. It's entitled The Use of Blood Collection Cards for Assessing Presence of Microcystin in Marine and Estuarine Birds. We had a few objectives here. I'm going to try to quickly go through this paper so that we have time to chat about our new stuff, but we wanted to determine if blood cards could be used as a screening tool for microcystin in live birds in rehabilitation. So these are, you'll see a picture in a bit, but there are these little cards that are really easy for rehabbers to use. We wanted to determine if those blood cards could also be used in conjunction with standard Abraxas microcystins ELISA kits. So we didn't know if they would work for these commercially available kits or if we needed something different. And we also wanted to determine if birds in Monterey Bay were carrying microcystin between 2011 and 2015. And if so, we wanted to know which species. So quickly, the results, the Abraxas kits did work. There was a clear difference between spiked blood and control blood, but we ran into this issue where for a conservative amount of blood, maybe 0.005 mils to be taken from a live bird, the lowest detectable concentration of microcystin was 31.2 ppb, and that's a pretty high level of toxin. So we're going to lose this resolution for smaller values. So you can see here on the right, this is one of the blood collection cards that I've been talking about, and the 4 spots combined here are 0.005 mils. So that was what our samples looked like for birds coming into rehab, and this is a really realistic amount of blood to be drawn from a live rehab bird. So we also found that to detect microcystin in bird blood at a realistic level, maybe 0.5 ppb, the blood sample would have to be at least a volume of half a mil, and that's a lot of blood for a live bird. So how we got around that was that we binned our samples. So the blood samples that we got from rehabilitation centers for the study were binned so that we could get that appropriate amount of blood. And what showed up here for us is that 2 groups tested positive for microcystin. Those were ducks, including surf scoters, and then also common murres. So this is the link to our new work. That 2017 paper let us know that microcystin could be an issue for surf scoters and common murres. And now we really want to know more about the historical and present potential impacts of microcystin to nearshore feeding seabirds in California. So I will come back in a little bit, but I'll hand it off to Rohini, and she can talk about the methods. And she did a lot of the methodological work. So it's really nice to have her here. So go ahead, Rohini, whenever you're ready.
Next slide. Hi, everyone. Thank you so much for that introduction, Corey. So I'm here to talk about our new work. So kidneys and livers were collected from surf scoters and the common murres during routine necropsies at the California Department of Fish and Wildlife Marine Wildlife Veterinary Care and Research Center. The kidney samples were taken in 2008, 2009, 2018, and 2019. And the liver samples were collected between 2006 and 2008 and in 2019. These tissues specifically were taken because it had been determined that they were reliable for toxin uptake when it came to domoic acid and saxitoxin. So now we're looking at microcystin. These are primarily Monterey Bay Area birds. And the reason for that is that microcystin had been detected in the coastal ocean there. So these birds were a good species to look at. And once those tissues were taken, they were kept in -80 and available for analysis further on. Next slide, please. So to extract the microcystin, the tissues were thawed and put into falcon tubes with acid methanol, which they were sonicated with. If the tissues were under 1 gram, they received 5 milliliters of acid methanol, and tissues that were at least 1 gram or over received 10 milliliters. Once sonicated, they were centrifuged for 20 minutes, and the resulting liquid we got was separated into 2 chlorophyll tubes. 4 milliliters went into one tube, and the leftover liquid went into another tube. Both these tubes were then placed in the freezer until they were cleaned at a later date. Next slide, please. So our cleaning methods, before any cleaning could take place, the 4 milliliter samples we had aliquoted were diluted to 100 milliliters using 96 milliliters of acid water, and that was put into these jars. On our vacuum manifold, we had done this kind of column tower setup where there was a Baker bond 500 milligram SP column attached to a Phenominix Strata X 100 milligram column, and then that was attached to the vacuum manifold where the pressure was kept constant at 5 inches of mercury. To clean these samples, an adapted protocol was followed from these 3 papers that I've listed down below, which all talked about microcystin extraction from saltwater mussels. So the protocol was adapted from there and applied to these bird tissues. 2 milliliters of eluted liquid was our result, and that was aliquoted further. 1 milliliter went into an LCMS vial, which was then put into a mass spectrometer for further analysis, and the rest was stored at 6 degrees Celsius. Next slide, please. So I'm going to talk a little bit about the microcystin variants that were found. All of these microcystin variants were tested for by Dr. Peacock in her lab. And a lot of these variants, they're important to talk about because there are over 200 different variants of microcystis that have been identified over the years. In a cyanobacterial bloom, you will have different variants of these microcystins. So knowing what is in the water can give you a good idea of how that correlates to what the birds were consuming at that time. Previous research has described MCLR as one of the more common variants, but it's also been seen that MCLW and MCLF may actually be more toxic than MCLR. All of the variants are important to consider because they all have relative different levels of toxicity. All of these variants work by targeting protein phosphatases, which are enzymes that are part of the regulatory cellular processes in the bodies. So that was a really interesting thing to see, was the different variants and how the different levels that they showed. Next slide, please. So this is just kind of a broad overview of the different results that we got specifically when looking at the variants. Corey will go over the results we got when just looking at MCLR. But in general, the highest amount of microcystin that was found was 2,454.23 nanograms per gram in a surf scoter, which is very high. There were more instances of MCLR and DMCLR in common mirrors than surf scoters. That was a really interesting result to see, mainly because of the differences in diet, which Corey will go over in a later slide. On average, there were higher levels of DM-MCLR than MCLR in each bird. This was also interesting to see, and the reason for this could be attributed to the thinking that although DM-MCLR and MCLR have similar toxicity profiles, DM-MCLR may be more stable in the environment, so it may be there longer for the birds to ingest, and that might be why it showed up more often or at higher levels than the MCLR. And overall, there was a low occurrence of MCLW, MCYR, and MCRR, and there was a comparatively higher occurrence of MCLY, MCLA, and MCLF. And MCLF is one of those more toxic variants when compared to MCLR. So it was interesting to see this difference of a low occurrence of some less toxic variants and maybe a higher occurrence of more toxic variants. And with that, I will turn it back over to Corey.
Hey, thanks, Rohini. That was really cool. So yeah, I just wanted to talk a little bit more about the specific samples. So we had both liver and kidney samples, as Rohini mentioned. And on the left here, you'll see a graph for those liver samples. We just looked at MCYLR for now. These are just preliminary results, as I mentioned before, so we can dive in a little bit more. But just to take a look at these graphs, on the y-axis, we have MCYLR in nanograms per gram. For the liver samples, it goes from 0 up to 2,200. For the kidney samples, it goes from 0 to 350. So if we're just looking at the liver samples, these are run by Misty. She had 282 samples, so a nice solid range. The range of data for common MERS, which are in blue, was between 0 and 2014. And I put the individual data points on here just so you can see that there's just such a wide variance between individual birds. Of course, that makes sense from other things we've looked at, encounter rates, and just individual variation in general and how they are going to take up toxin. So yeah, for the common MERS, it was quite variable and quite high. And then the surf scoters were also variable and high, but they had a little bit fewer samples, so 100 instead of 182, but the range was sort of similar, so 0 to 2097. So that was interesting to us, and I'll go into why we were actually a little bit surprised by that information in the next couple slides. But let me talk about the kidney samples also. So of course, it's a hepatotoxin. You would expect to find it in liver samples, but as Rohini mentioned, kidney samples can be really helpful too sometimes, especially if something the bird's not in great condition, or you can only get kidney or just as something to back it up. So there was 25 kidney samples run. 15 of those were common MER and 10 surf scoters. And you can see here that the ranges were 0 to 32.46 for the common MERS and 0 to 286 for the surf scoters. And they also had a lot of variation. I think what we're seeing here with the smaller range of common MERS is probably just an artifact of sample size, but they may have just take up less in the kidneys as a species as well. So something to think about. But let me get to the next slide. Okay, so Rohini sort of touched on this earlier, but the diet for these birds is different. So we were actually pretty surprised to see common MERS be so high in those liver samples. Again, variable, but some of them were pretty high, as high as surf scoters. So their main diet is fish, krill, copepods, and squid. But in California, they mostly eat northern anchovy. The surf scoters, we were not as surprised to see, and their diet is clams, mussels, fish eggs, sometimes aquatic vegetation, but the largest part of their diet is shellfish. So when we're looking at what we know about how those prey items take up toxin, here's a paper that Misty, Rafe, and I did way back in 2016, and this is these are tank experiments. We had low tanks on the left and high tanks on the right. And you can see here just an example that these are California mussels that surf scoters are likely to be eating in the Monterey Bay. They do take up toxin immediately in 24 hours, and then they hold on to it. You can see on the x-axis here, it's hours to weeks, but they hold on to it up to 8 weeks. So, you know, the encounter rate for surf scoters could be quite high based on that. And if the toxin is higher, you can see here on the right side, they uptake toxin and then they depurate it a little bit more slowly when the encounter rate's higher. So yeah, again, just supporting that surf scoters encountering prey items with microcystin in the marine environment is not that crazy. We don't have as much information for the northern anchovy. I did see, Misty, that you had a couple of northern anchovies that you tested. I'm really curious about your numbers for those. And I know that there's probably no dose experiments going on for them, but it would be really helpful here. But in lieu of that, we have this one study where they looked at saxitoxin and microcystin. Saxitoxin is in the gray, microcystin is in the darker bars. And they looked at killifish on the left, and they looked at tilapia on the right. And they're looking at muscle and viscera for the killifish, muscle and liver, microcystins for the tilapia, and along the x-axis, they have their depuration groups, which were 8, 16, 24, and 32 days after field collection. So these were fish collected during a bloom. And they really held on to toxin, as you can see here. And the main take-home point from their study was that the cyanotoxin depuration appeared to be a species and tissue-dependent process. And of course, there's a lot of other studies on microcystin in fish, but not ones that I know of for northern anchovy, most likely because they're a marine fish and we're talking about a freshwater toxin. So Misty, I'd be really curious to see what your numbers look like there. So another thing to think about here is what are the effects of these types of numbers on birds? And that's also hard to figure out. So the standard toxicological endpoint, such as like LD50 for oral route of exposure, is sort of lacking in birds. There have been a couple of studies where they've experimentally induced mortality for birds by injecting into the intraperitoneal cavity. So that's into the body cavity rather than an oral exposure. On the other hand, we do have a mammalian oral dose, LD50 value, and that's 10,000. So these, in this study, the authors, they did some dosing and they came up with what would it look like for a quarter LD50, half LD50, half the LD50, regular LD50, I'm sorry, and then double the LD50. And what would that look like when we're talking about damage to liver in birds? So you can see here, at the quarter LD50 dose, that's about 2,500 micrograms per kilogram of microcystin, that they're starting to see some mild damage in those birds. And so that 2,500 value, that's sort of similar to what we saw in our liver values. But I want people to keep in mind here that the microcystin that birds are encountering in the environment, it's likely to be higher than what's going to be in the tissue values. So there's some microcystin values that are available in the literature. There's some for ducks, and there's actually quite a few for flamingos. And some of those are quite high, but in some cases, our results are much higher than those that are reported. And if people have questions about that, this one paper did a little bit of a review of like what's known about values for birds and what killed each type of bird. So the ranges really vary between species and location for microcystin when it comes to birds. But we took this and we thought, okay, this gives us a little bit of a basis of what kind of harm these values could be causing. So our thoughts and next steps. So results, as I said, were quite variable. They varied by individual, much like other in situ bird and shellfish samples. Northern anchovy and other forage fish might be holding on to microcystin toxin longer than anticipated in the marine environment. So it'd be interesting to see how other marine fish are acting or reacting to microcystin and how they're holding on to toxin. We don't completely understand the toxic thresholds for birds and microcystin. Is it species specific? It seems like it may be in some cases, but we do have some sort of idea of what level causes damage in some species, and our values showed that birds were likely exposed to damaging levels of microcystin. We still have a lot of work to do on this, and we want to dive deeper, both temporally and spatially. We want to try to line this up with pulses of microcystin in the nearshore marine environment in Monterey Bay if we can. We'd like to dive into necropsy data to examine correlations between growth signs and those values we got for kidney and liver. And then we'd also like to compare to ELISA. So even though it's been documented that ELISA and LCMS are both capable of accurately detecting DA and comparable across methods without false positives, we know that it can be helpful to managers who use ELISA more readily than LCMS. So that's some of our next steps as well. So with that, I know we're probably at right around time, but we'll take any questions.
Thank you so much. Very interesting work. If we do have questions, let's put them in the chat. We're, just at time, like Corey said. And also feel free, of course, to email her. And yeah, thank you guys so much for joining. I really appreciate it.
Yeah, thank you.
Okay, and on to our next presenter. So Zane is here from Water Boards. He's going to give us a little bit of an update on the cyanotoxin provisions and that project that he's working on. So go ahead, Zane.
Oh, thank you. So yeah, so I don't have any slides. I just really quickly wanted to update everybody on our progress on our project that we're working on. So previously the project had been, the name of the project was the Biostimulatory, Cyanotoxins, and Biological Conditions Provisions. And so a lot of the provisions focused on the biostimulatory and biostimulatory effects and also on biological conditions. And as we've met with various regional boards and other stakeholders, we got the input that the focus really needed to be on cyanotoxins and harmful algal blooms. So having taken that input, we went back and we revised the project charter and the focus on goals of the project. And so the project now is the harmful algal blooms and cyanotoxin provisions. So it's really focused on those two elements. Not that the other ones aren't important, and we'll focus or incorporate them as they pertain to harmful algal blooms and cyanotoxins, but not focusing specifically on those other factors. So right now, we've updated our project charter and we're briefing the board on the new project charter I'm showing you. Subsequent to that, what we plan to do is to host 3 outreach meetings, focused outreach meetings on specific groups that we want to then sit down and share with them the project charter, and we go through that with them and the project goals. The first group that we want to share with is the tribal groups. So reaching out to all the tribes and tribal representatives that want to interact with us and discuss this with us. The next group that we want to reach out to is environmental justice and the environmental groups. And then we want to hold a third outreach meeting with all of the dischargers, including lakes, owners, and operators. So we're trying to really go out and get lists to reach out to all the various groups. But that's a big undertaking, and so if you're concerned that we will miss you, if you really want to attend or know people who really want to be part of this, I encourage you to then reach out to us. I'll put an email link into the chat, and you can email that or email me directly and make sure that that we send the invitation to that group. And when you put email, please split with affiliation with a sort of the tribe and justice group of this tribe. And we'd really like to know that, so we can invite you to that meeting. So that's the next big step, is to host these meetings. Again, we will introduce and go through the charter in detail and what is or isn't being included in this project, and let you have that discussion. And part of that can inform what we're doing on this project, as well as inform us and what we're doing in subsequent projects. So we envision that we'll continue to work on this into the future. And we also, part of that meeting will be to discuss what we're calling our statement of necessity. So we really, one of the things when we do revisions, we need to go through and explain why this is a big issue and what's going on within that. We have a lot of that information, but we'd love to, as part of that, be able to take a lot of input on how different groups and people are being impacted, some of the more local impacts. I think that's going to be really telling as to how it's impacting local economies, um, health, recreation, things that are going on a local level. What is impacts of being seen by people at the community level. Part of that is also the economic impact. So we need to do, as part of any provisions, we need to do an economic analysis. Usually the economic analysis allows you to focus on what it costs to achieve compliance. But it's also important to understand what the cost is for not achieving the compliance. So what's the cost for not being able to keep, to dollars coming into a bank, for example, because they're not able to stay open because of the impact from the labs. Subsequent to those meetings, then, what we want to do is we plan to host a public workshop. And at that public workshop, so we still have a bit of work to do. One of the things we're doing is we're taking a very close look at the different cyanotoxin thresholds that are out there. So the CCHAB network obviously has thresholds for caution, warning, and danger levels. With Health Organization, US EPA, and we have all put out levels and science. There's some both those levels. And so we're reviewing that. We have a contract with them also to really go through and do that. So once we have that, then we'll come to a public workshop to share that information, as well as the developing the narrative objective for clinical algorithms, and we'll to share that, and along with what we're thinking for a program of implementation and also for a monitoring. So those are some of the things we look forward to when we get to posting a public workshop. And that's, oh, and then the other thing is, so we're hosting these larger groups. If you're interested in talking to us more in a one-on-one, we're always open, and you can email us and ask for us to meet that one-on-one with us, and that is happy to try to groups, to have a discussion about what we're doing. So that's my update. You don't have any questions?
Yeah, we do have a minute or two for questions, if anyone has any.
Hey, Zane.
Yes.
Question on the workshops you were talking about. I might have missed it. Did you mention the timing of when you're planning to do those?
So we're hoping to, in the next several months, so this kind of early second half of this year, to be able to do these outreach meetings.
Okay.
We don't have them exact right on the schedule at the moment. We're still going through our board briefings.
Gotcha.
And we'll start to set them up as we're putting together the list of invited. I think our biggest challenge is getting a complete list of people to invite.
Gotcha. Okay, so the invite's gonna go through you're gonna create a list as opposed to putting it through a listserv, because that was my follow-up, was where would people keep an eye out for it?
Yeah, so we'll send a list and send that out.
Okay.
We might use a listserv for the tribes. So there's a tribe listserv, but and then yeah, so the workshop, and come to a public, open public workshop, and we'll discuss some of the details. That's maybe this year, but likely next year.
Thank you.
Sarah?
Hey, Zane. This is Sarah with Big Valley Rancheria, Big Valley Band of Pomo Indians, and this is great that you're working on this. We're definitely interested in being involved, and boy, there are a lot of tribes working on cyanotoxin monitoring. Just seems to be growing over the years. So I think it's great that you're going to be doing some outreach. We'd love to assist with that. Question I have for you, you mentioned a public workshop on the cyano thresholds, and I just wanted to clarify. So is this is what the state looking at is, whether those thresholds should be updated in any way based on, either federal or other just other thresholds that the state might want to adopt, and it is ultimately to adopt not just suggested action levels, but to adopt the standards themselves.
Correct. So that's there's several different thresholds right now. And so what we need to do is choose which one would we use for a water quality objective. So it does have, more, yeah, it's not just a recommendation at that point, it would actually be used for making listing decisions. And so it becomes a little bit more regulatory.
You're looking at drinking water as well, or is it just Rec 1, Rec 2?
We're looking at recreation and drinking water. So those are the two.
Okay.
Focused on at the moment, yes.
What about fish consumption?
Not at the moment, yeah, and this is part of some of the discussion that occurred during the outreach meetings is, yeah, which ones looking at that, yeah. So part of it is, we're looking at where do we think we have the science to support it, and where do we think we need additional work, and that's part of the discussion we want to have is, okay, what, we're going to take these on now. We're also continuing to gather information on additional parameters. So the ones, the great presentations today on, what's going into shellfish and what's happening to wildlife and what's happening to aquatic life. Like all those ones are water quality objectives we'd love to protect, that we need to have the science enough to do that. So, which ones do we take on next? And then, and part of that is we determine which ones can you actually get enough science to support. There's a lot of concern about dog deaths, and a lot of concern about tribal cultural practices. All those things are important and being considered.
Great, thank you so much.
Thank you.
Anyone else? All right. Zane, I wonder if you would consider also doing a similar workshop for the CCHAB group to weigh in as well. I know a lot of us are interested. Not all of us are part of one of those groups.
Yeah, I'd be happy to come back to a future CCHAB workshop meeting and
cool.
present.
That'd be great. Thank you.
Yeah.
All right, important stuff you're doing there. Okay. And last but certainly not least, we have Jamie, our very own from SWRCB, who's going to be presenting for us. Jamie, do you want me to share, or are you good, Karen?
Should be good to share. Hopefully, let me
give me know you have issue.
Get it in the right. Let's see. Are you seeing it in presenter or?
I am seeing presenter view.
Perfect. All right.
Oh, sorry. Just not the presentation itself, but like what with the notes and everything.
Oh yes. So let's switch it.
Share screen.
This is supposed to. Oh, this. Hold on. You'd think that after many years of doing this, better at this, but how about now?
All right, that looks great. Thank you.
Okay, thank you. All right, so I am following on with the bioaccumulation theme here, and so Corey and Misty set me up really well to talk about some of the work here that we did in Southern California. And so before I get started, I just really want to acknowledge two folks. There were many folks that worked on this, but Allie Lee and Sam Lillywhite did a big lift on all of this. Sam did a ton of field work, and then Allie did a ton of lab work and bioinformatics and toxin analysis that brought this all together, as well as assisting with a lot of the data synthesis. So I wanted to thank them both, and I'm presenting on behalf of all of our work here. And so I'll just start off real quickly. Again, we've talked a bit about this already, but the motivation for this work is there's a growing body of work, and Missy showed a map, really demonstrating how this happens in many places throughout the United States, and particularly there's growing documentation of this in California, where harmful algal bloom toxins move through the watershed down into the ocean. So as they say in Finding Nemo, everything goes to the ocean. And so we know that on the inland side that we're seeing more and more freshwater harmful algal blooms. And so that kind of increases our risk for the presence of freshwater toxins at land sea interface locations. In addition, I know we're a freshwater group, but we have heard from, from time to time from our marine partners We've had several years of very intense domoic acid blooms on the marine side. And so, this zone is really a hotspot for multiple toxins. Risks to recreation are really not well understood, but we've seen that it bioaccumulates in many different species, all the way up to birds and even marine mammals at some points. And so, a lot of the work has been site specific. And so the goal here was to kind of expand geographically the knowledge of where and maybe when this is happening. And so the goals of this particular work, which I'm going to say is this is kind of we're in progress with this. So I'm going to talk to you about some of these objectives, and some of them are in progress, and maybe we'll come back at a different meeting and give you an update. But was to just look at the extent and magnitude of freshwater and marine toxins in the receiving waters in the LA and Ventura area, and look at the temporal dynamics in when and where you see multiple toxins, either marine, freshwater, or both, focusing on microcystins and domoic acid in the environment and in bivalve shellfish, and to characterize the extent that the toxin is correlated with the specific HAB taxa that we believe are producing them, as well as water quality conditions. So this was a field study that we did in 2024. We sampled across 11 stations throughout kind of the LA, Ventura area, and we broke these sites. We just kind of put them into the category of coastal sites. So a truly marine station that was a beach or a harbor, or a little estuary embayment type area with marine salinity. So we had 7 stations like that. And then we had 4 stations where we were able to kind of go a little bit upstream of those coastal stations and look at what we thought might have been, quote unquote, our source location. So Ventura, Santa Clara River, Malibu Creek, and Leo Carrillo Creek as freshwater systems that flow into directly the marine system. We focused on domoic acid and microcystin characterization, as I mentioned a moment ago. And we looked at multiple kind of indicators of toxin. So we looked at toxin accumulation in bivalve shellfish, focusing on mussels. We also use SPAT passive samplers, and I had a great intro as to what those are. But if you miss those talks really quickly, those are essentially a time integrated passive sampler that can passively adsorb and desorb algal toxins, multiple algal toxins. So it's really getting at what is in the dissolve phase over time. So it's essentially kind of like an artificial chemical sampling mussel that is not alive. We also did direct water grabs for algal toxins. We looked at eDNA to look at some of the algal taxa that were present and at the same time as these toxin indicators, and did some basic water quality characterization. So chlorophyll, total nitrogen, total phosphorus, and temperature, salinity, conductivity with a field sonde. And so we visited all of these sites on a monthly basis for a year to kind of get an annual cycle of when you see toxins and what their corresponding environmental correlates were. So the field and lab sampling process was pretty intense. Again, it was a team of people. Here's some of our lab assistants at SWRCB. I'll just call them out, but we have Thomas and Shannon and Jared here who did a lot of work with us. And so we collected bivalves from ambient beds. So we were just collecting from, off of rocks or off of docks where the mussels were existing naturally. We weren't deploying any sentinel mussels. We collected a minimum of 10, sometimes a little bit more to make sure we could get enough mussel mass. They were returned to lab, shucked and homogenized within 48 hours, typically actually shorter than that, typically either the same day or the very next morning. Turn them into a lovely mussel smoothie. And then the tissues were cleaned up and analyzed via LCMS for microcystins and domoic acid. We also did the spat bags. So those are deployed as you do a spat bag. A water grab sample that we filtered water down onto a filter. So essentially a particulate measure of domoic acid and microcystin. filtered water down for eDNA, and like I said, the basic water quality parameters. So just quickly jumping into the results. And again, we're working this up still. So these are some of our preliminary results here. This is maybe not a surprising finding given what we saw from Misty and from Corey, was that algal toxins were pervasive in the mussels across our various sampling sites. So here I'm showing, I know I said there were 11 sites. There we did 7 coastal sites, so we didn't have any mussels in the more fresh upstream sites. So these are all of the sites where we had our ambient mussel beds. So again, these are just a lot of these we were walking up to rocks or climbing on, going off the side of a dock and pulling mussels off of those environments to do this. And so on the left-hand panel, we have domoic acid. So I don't know if you can see my cursor, but on this side, this is domoic acid. On the right is microcystins, and we have a heat map here. So anything that is gray is below the method detection limit. And then if you start getting a blue up to a red, those are detectable levels. And these are both in parts per billion. And so just as a reminder on to orient you as to what these numbers kind of mean, domoic acid has a federally recognized safe to eat guideline of 20 parts per million. So on the domoic acid side, these are in parts per billion. So these are all, even though this is we have a red up here in Tar Harbor, that's the study maximum that is not exceeding the FDA limit. However, these are pretty far below. However, the OEHA and WHO do have a safe to eat level of 10 ppb guidance. And so we do see a number of samples here, anything in the darker blue or higher is exceeding that OEHA WHO level. So we're seeing on the microcystin side as well as the DA that algal toxins are very commonly detected in mussels. And then when it comes to microcystins, they're very commonly over that safe to eat level throughout the sites. And there's some interesting kind of temporal dynamics that we see here with where we see higher and lower concentrations, with some of the higher concentrations coming in the earlier months for microcystins as well as for domoic acid. And then to summarize, again, so you can kind of see everything together, so you don't have to kind of stitch it together in your mind. Here's just an overview of where we were seeing toxins in a presence-absence standpoint, where we were kind of looking at co-occurrence. So the red color is where we have both toxins present at that location. Blue is domoic acid only, green is microcystin only, and then gray is no algal toxin detected. So if you notice on this plot, there are very rare instances of no algal toxins detected. There are a few here in February. That's the month where we had the most common number of samples with below detection, but that is also a period where we had a lot of rain and a lot of flushing. So we're unsure if that is related to that trend, but we're looking into if that's maybe a contributing factor. But co-occurrence of algal toxins in mussels in the Southern California area and LA Ventura area is very common. And so I mentioned that we were measuring toxin in a variety of matrices. So this is water detection of domoic acid and microcystin. So again, on this plot, we're looking at domoic acid on the left and microcystin on the right. We did water testing in the river sites as well. So those the sites that we believed were influencing our coastal sites. And so those are added back on. That's why there's more sites in this figure. But the takeaway here is that you see a lot more gray. There are some intermittent detections of domoic acid. Very rare for microcystin. Actually, none of the coastal sites have it, only the Santa Clara River in the later part of the year. If you've ever been there, it's very green around this time of the year, so there's a lot of biomass there by the end of the fall season. And so very rare detection. And then just to pop this up so you can kind of cross compare, very rare detection of toxins directly in the water and particulate matter. Using our methodology, we use the ELISA for our water samples and filtered a couple hundred mils down, but much more common detection in our bivalves that bioaccumulate. And so our third way that we were looking at toxins is with spat. And so spat aligns a lot more with our tissue observations. It's telling us a story of more frequent detection. You will notice that there's some white spots. That's because we lost some samplers. People like to steal them. A lot of these are really commonly frequented areas. And then Leo Carrillo dried up at a certain point. So we stopped sampling at this location in July. So a little bit harder to get these out consistently at all the sites, but I think what we still see from this indicator is that toxins are a lot more common. And again, showing domoic acid on the right, domoic acid on the left, and microcystin on the right. And so when we look at statistically, spat does correlate well with the water and the tissue domoic acid. And interestingly, we don't see any significant correlations with microcystins, even though we do see a bit more prevalence in the dissolve phase. It doesn't significantly correlate with microcystin, and obviously because we had no detections with water, mussels didn't correlate with water either. But spat, again, tells us a pretty similar story of frequent toxin co-occurrence across these locations. And so that was one of the things that we sampled. We also looked at algal community composition, looking at both the 16S to look at cyanobacteria and the 18S to look at our eukaryotic marine HABs. And so we're in the progress of doing this. So we're showing just the first 6 months here. So the take home here is you're seeing not a lot of, this is what we're showing here is we isolated out the potentially microcystin producing taxa. And so we're getting a lot of non-detects or very low. This is the percent of reads. So our max scale here is 30%. This is the Ventura River. This is Leo Carrillo Creek. And this is actually a marine site, Alamitos Bay down in Long Beach. But for the most part, the rest of the locations, we're actually not really seeing a lot of toxin producing taxa. So we aren't seeing good correspondence with the producers in most locations, at least for the first 6 months of our study, when we saw those higher microcystin levels in the mussels. And what also was interesting is the domoic acid producing taxa on the marine side, Pseudonychia, is also, from a relative abundance perspective, not particularly a dominant in the community. This is that same plot. These relative abundance numbers are actually less than 1%. It is detectable though in more sites, which aligns a bit more with what we were seeing with the toxin domoic acid being present much of the time. And then the other thing that we saw that we thought was an interesting finding, and we're still digging into this, is that we saw a lot of other potentially harmful marine taxa throughout the year. So that the critters are here, so a lot of dinoflagellates and even some rhaphodophytes present on a routine basis throughout the first 6 months of the study. So they're pretty prevalent in some locations. For example, a lot of Akashiwa, which is a surfactant forming dinoflagellate, was present a lot in June. So we're still working on this analysis, but I think some maybe take-home messages and points is that we saw a lot of evidence, and again, this is maybe not super shocking, having heard Corey and Misty's talk, but microcystins, our observations really point to frequent microcystin transport into the marine environment. Microcystins were commonly observed throughout the year in most of our locations. We saw some intensification in some places. It was persistent in the dissolve phase. We saw it a lot in the spat, very, very rare in the particulate phase. And we also saw rare taxa that produce the toxin in the river and coastal sites. Again, suggesting that the toxin itself is transporting, not that the cells are in Southern California in this region are moving and producing toxin kind of in the environment themselves. And so to kind of wrap things up, HAB toxins, marine and freshwater drive, are very present, very commonly detected in mussels. The magnitude varies quite a lot. And so this is something where, continued kind of surveillance of the microcystins is probably something that would be good to do to really understand, the potential impacts of this. The co-occurrence is also frequent, and the human and wildlife health implications. I know the domoic acid was typically well below your acute exposure levels, but it was present a lot of the time. So what does it mean to take a really low continuous dose of a neurotoxin if you eat a lot of wildly harvest shellfish? We don't know. And we're currently working on finishing out our eDNA analysis and our analysis with other variables. So that's coming soon. And I just wanted to give a little plug for, we have a complementary study, which is the Southern California Bite Regional Monitoring Program that was done at this same time. And this was a larger bite-wide, so all the way from Santa Barbara down to the Mexican border, look at algal toxins in bivalve shellfish, looking at both mussels and oysters. In addition to some other indicators, looked at viral pathogens, microplastics, and other contaminants like heavy metals at the same time as the algal toxins. And that's going to all come together into a risk assessment analysis to look at bioaccumulation of a variety of contaminants throughout the Southern California Bight. So that will be coming soon. And if there's interest in that, we would be happy to come back and talk about the results of that work. So with that, I will stop talking, and I'd be happy to take any questions if there's time for questions.
There is time. I thought Ali was answering some questions in the chat, but if anyone else has questions, please speak up. All right. I'll always feel free to ask Jamie. You know where to find her.
I'm standing between people and lunch, so I get it.
Yeah. So a couple announcements just before we go here. So our next meeting will be August 12th. And again, 9 to noon as is standard. So we'll see you then. Also just wanted to do a shout. If you guys have ideas for future presentations, please let us know, or themes you'd like. We're always interested in hearing and what you guys are interested in and tailoring it to what our interest group has in mind and is thinking about. And if you have a project or something you want to present on, just reach out to us. Also, I saw that Zane put in the chat that if you want to be part of those outreach meetings for the cyanotoxin provisions, he put an email address to email. So I'm just going to repost that in the chat. So it's at the bottom there. You guys can grab that. I'll leave this open so you can have a second to grab it. And otherwise, yeah, we'll see you August 12th. And thanks so much for coming today.